Hi, in this video I'm going to talk to you about gene expression. We're going to talk about transcription and RNA processing. This is the first part of gene expression and it happens in the nucleus. Take out a sheet of blank paper, so just regular old printer paper, and you're going to fold the printer paper in half, hamburger, and then you're going to fold it in half, hamburger again, and then you're going to fold it in half, hamburger again a third time. Once you're done folding this paper in half, it should be about that big and it should be small enough to fit in your pocket. Then what you're going to do is you're going to open it up and you'll notice you have 4 spaces across the top and 4 spaces across the bottom. I would recommend that you use each one of these spaces for each one of these slides that we're going to talk about. So for this first one, you're going to write gene expression, transcription, and RNA processing. So this is the first part of protein synthesis. So let's get started. Let's see. All right, first we're going to talk about transcription. Let's define this process. So transcription, let me get my black ink. Transcription is a process. It's a process of using DNA to make RNA. Now you can make all different kinds of RNA, but in this particular process that we're talking about, we're going to make messenger RNA. This process happens in the nucleus. All right, so that's it for your second square. So your first square is the previous slide, second square is this slide. The third square, we're going to talk about the promoter region. So the promoter region, in order to describe what the promoter region is, we should draw a molecule of DNA. So DNA is double-stranded. So this is one molecule of DNA. And we're going to draw the promoter region. The promoter region is a location, or it's part of the DNA. So the promoter region is here. We're also going to draw in the gene. The gene, in this case, if we trace that same strand, the gene is going to be down the DNA strand from the promoter region. So this is the gene here, whoops, excuse me, and this is the promoter. So let's define this. So the promoter is a DNA sequence where proteins bind to initiate transcription. Now, a promoter region is anywhere from, let's say, 100 to 1,000 base pairs. And these base pairs are upstream from the gene. So you can see what we're talking about here. The gene is here, and if you go up from the gene to the promoter, you'll see that the promoter is what's called upstream. So when we talk about this, we're talking about upstream and downstream. So there's sometimes you'll hear this term called a Tata box. A Tata box is an example of a promoter region. So a Tata box is a thymine, an adenine, a thymine, and then 3 adenine bases. So this is one one of many promoters or promoter regions in eukaryotes. And that's what you need to know about promoter regions. So there are 3 parts to transcription. The first part to transcription is initiation. So this is the first part of transcription. And we'll describe what this is in just a minute. So again, we need our DNA molecule with its 2 strands. And we've got our promoter region here. And we've got our gene down this way. It's kind of hard to see over the black, so what I'm doing is trying to kind of color that in a little bit. So we have our gene, whoa, kind of getting a little offline there. So this is our gene. It's supposed to be a single line, but you get the idea. So this is our gene, and we've got our promoter. So what happens in initiation is there's this molecule called RNA polymerase. There are several types of them, but the RNA polymerase binds at the promoter region. When it binds at the promoter region on the DNA, it allows for another molecule to come into place, and this other molecule starts right here. This molecule is helicase, and it's actually called DNA helicase. Sometimes we just call it helicase for short, and helicase has a couple of functions, and we'll talk about what those are. So in initiation, part 1 of initiation is DNA, oops, excuse me, let's cross that off, RNA polymerase binds to the promoter region on the DNA. The second thing that happens in initiation is DNA helicase uncoils the DNA, or the double helix, oops, double, and it also breaks the hydrogen bonds between the bases. So this is initiation. This gets the process of transcription started. So there are additional parts. There we go. The next is elongation, and that's exactly what it sounds like. What we're going to do is we're going to begin building an RNA molecule. So in order to do this, what we need is we need an open strand of DNA. So we've got this open strand of DNA, and again at one end we have the promoter. So our promoter is here, and we've got our gene, and our gene is here, and our gene goes all along here, all the way over that way. So this whole entire strand is our gene. So our gene starts here and goes all the way that way. So you'll notice I keep referring. I want to make sure that I get everything in this picture for you. So we're also going to draw another molecule. This molecule we're going to draw right in here. So it's going to be drawn just like that, and it falls out of the DNA. So this is our pre-mRNA. So our pre-mRNA is here. So we're going to read the DNA in that direction. Since we're reading in that direction, this is the 3' end and this is the 5' end. Since this is the 5' end, this is going to be our 3' end of the pre-mRNA, and this is going to be the 5' end. So what happens is this pre-mRNA is being built by this molecule that takes up this entire area. This is called RNA polymerase. And that's what this big molecule is right here. So what happens is RNA polymerase is around this entire area. We have an area that's open here in a bubble. So let's describe what's going on here. So this is the second part of transcription. And in this RNA polymerase, guides new nucleotides into place and covalently bonds them. So it'll bond that new nucleotide to the growing strand, and it's binding it in a 5' to 3' direction. Now, all the while, it proofreads the strand. So again, we are adding nucleotides at the 3' end. So don't let that mess you up. All right. So things to remember on the next slide. What I want you to do is actually on your next space, what I want you to do is make a note. So let's see if I can squeeze this down here. So I want you to remember this. If we have A, T, G, C, that DNA would go with T, A, C, and G. These are both DNA. If we were going to use this DNA to transcribe, we would have U, A, C, and G. This would be the this would be the mRNA. Okay, that's what that would be. So I've got a little squiggly there separating those. Now, this top strand, this may sound really weird, but this is the coding strand. The coding strand is not used to make the mRNA. It's called the coding strand because if you look at it, it's exactly the same as the mRNA, except for the T's turn into U's, or the T, instead of having a T, it has a U. This is called by several names. It's called the template strand. It can also be called the non-coding strand. It can also be called the minus strand. It can also be called the antisense strand. All of these names are all talking about this strand of DNA that is actually being used to make the messenger RNA. So this is the template strand, non-coding strand, minus strand, and or the antisense strand. This up here is called the coding strand, which feels a little odd. All right, so termination. Termination is the third part of transcription. And termination is just simply, it's a termination sequence on the DNA that is arrived at by the RNA polymerase. And that's all termination is. Termination is pretty straightforward. So that one's pretty short. So those are the three parts of transcription. Now we've got RNA processing or RNA editing. So basically what we've got going on here, we've got a long molecule. This is a pre-mRNA. So it's not quite ready to be called an RNA. So if you take this and you divide it, I don't know, let's go 1, 2, 3, 4, 5. Let's put 5 marks on there. So on a pre-mRNA, we have introns, or we have an intron, that's from here to here. That's this portion of the mRNA. We have exon, and then we have an intron, and then we have an exon, and an intron, and an exon. So then we go through this process called RNA processing or RNA editing, either one. And the molecule that does this is called a spliceosome. And what a spliceosome does is it cuts in these locations. You won't be able to tell where these locations are, but the molecules in the cell can. So RNA processing gives us, uses spliceosomes. So what happens is we take this exon and we keep that exon, and then we splice to that the next exon, and then we splice to that the third exon. And then what we do is we add to each end a different molecule, or excuse me, an additional part. So if this is the 5' end, this would be the 3' end. And on the 5' end, there's a GTP cap that is put on this end. This is a guanosine triphosphate. And this is used for recognition by the ribosome in the next step. We'll talk about that next time in the next video. So over here we have what is called a poly-A tail. And a poly-A tail is numerous adenines that are tacked onto the end of this. What it does is it protects the genetic information from being degraded. So right here next to this 5', we have an A, we have a U, and we have a G. If you think about when we start school in August. The abbreviation for August is AUG. Well, that makes it easy to remember that AUG is the start codon on this mRNA. So this entire molecule from here to here is now an mRNA, and this is the start codon. The start codon is at the 5' end with the GTP cap, and at the 3' end we have the poly A tail. So the next thing that we'll talk about is how we use this mRNA to make a protein, and we'll do that next time.